And our last speaker this session is Seungsoo Kim, who's going to talk to us about the 3D architecture of the yeast genome.
Thank you to the organizers for giving me the opportunity to speak today, and to all of you for sticking around for the end of the session. We all know that the genome is comprised of several linear chromosomes, 16 in haploid yeasts, but these chromosomes aren't just laid end to end inside the nucleus. They're organized in a highly non-random and active manner inside the tiny space of the nucleus. And this 3D genome organization has important implications for nuclear processes ranging from transcription to DNA repair and recombination. Decades of microscopy have taught us a lot about the organization of the yeast genome. We know that one of its main features is that it's in a Rabl-like orientation. By this, I mean three things. So all of the centromeres are clustered together at one end of the nucleus because they're attached to the spindle pole, which persists throughout the cell cycle. The nucleolus, or rDNA, sits at the opposite end of the nucleus, and the telomeres tend to be near the nuclear periphery. More recently, we've used the method called Hi-C, a high-throughput version of the 3C assay developed by Decker, which uses high-throughput sequencing to measure the interaction frequencies between all pairs of loci in the genome. The data can be represented as a heat map like this, where each linear axis represents the linear genome, and the color of each pixel represents the interaction frequency for a single pair of loci. You'll notice two things about this heat map. There's a bright diagonal, which represents just the basic tendency for sequences right next to each other to interact in physical space. And secondly, there's this plaid pattern, which reflects the centromere clustering. Now, some have argued that there's not much more to the organization of the yeast genome because a simple polymer model, which tries to capture these basic constraints, is able to reproduce lots of different kinds of experimental data, including Hi-C, as you can see here. So that leaves us with the question, is there anything more to learn about the budding yeast 3D genome organization? But one thing that these previous studies haven't considered, of course, is diploidy. As Joseph mentioned earlier, of course, most wild strains of yeast are found in a diploid state. And in diploids, we know that from various fish studies that homologous loci tend to be found to be closer to each other on average than non-homologous loci. And this is even in mitotic growth without any meiotic pairing. Proponents of mitotic homolog pairing argue that this is the result of transient pairing of homologous DNA sequences, and they think this happens through a direct pairing of naked DNA. But others have argued that this is just a result of the Rabl-like orientation or of fish artifacts. In addition, there might be protein-mediated pairing as well, but we don't really have a conclusive answer to any of these questions because most of the studies so far have been limited to just a handful of genomic loci. Though, if mitotic homolog pairing does in fact occur, it could have important implications for DNA repair and various other processes. And so we wanted to look at mitotic homolog pairing genome-wide. And we did this using Hi-C and hybrid yeasts. You've also heard a little bit about some of the divergence between strains, but so Hi-C normally has a hard time distinguishing between homologous chromosomes. That's because it relies on sequence differences, and homologous chromosomes tend to be very similar. But of course, in yeasts, we can make hybrids between strains and even different species that are highly diverged, such as cerevisiae and bayanus, or also ovarum, which only have about 80% coding sequence identity. This allowed us for the first time to get a complete view of a diploid genome's conformation. This is what that view looks like. And this is for the cerevisiae bayanus hybrid. We also did this for other hybrids, but I'll just focus on this one for the sake of time. So here I've plotted the cerevisiae chromosomes followed by the bayanus chromosomes in homologous order. As before, we see the same diagonal signal, and the plaid pattern as before. Just to reassure ourselves that this wasn't any sort of artifact, we also did a control experiment in which we just mixed the two haploids. And here, we don't expect to see and don't see any pairing or interactions between a cerevisiae chromosome and a bayanus chromosome. Now, for the purposes of looking at mitotic homolog pairing, we want to focus on this quadrant up here, which represents interactions between a cerevisiae chromosome and a bayanus chromosome. The question we want to ask is, are the homologous interactions, which are along this diagonal here, stronger on average than non-homologous interactions? This is a little bit tricky to do, but the most careful approach we could think of was comparing each homologous interaction, or basically pixel on this diagonal, to the set of similar non-homologous interactions. And by similar, I mean that the two loci are at the same distance from the centromere and on chromosome arms of similar lengths. And this is to account for the Rabl-like orientation I mentioned earlier. So we can take these comparisons and aggregate them across the genome, and what we find then is an estimate of mitotic homolog pairing. So here I'm showing you those results for yeast in stationary phase, in exponential growth, and finally a control using the data from the Rabl polymer model that I mentioned earlier. This last set represents a sort of negative control because the polymer model doesn't directly encode any mitotic homolog pairing, so we shouldn't see any signal here. And so the fact that we see experimental homolog pairing that's greater than this suggests that, in fact, mitotic homolog pairing does seem to occur in cerevisiae bayanus hybrids. We hypothesized that maybe this homolog pairing might be stronger in hybrids where the two homologs are more similar, such as cerevisiae hybrids. But surprisingly, we found that the homolog pairing seems to be as strong in the cerevisiae bayanus hybrids as in the cerevisiae hybrids. In both cases, though, we see that it's stronger in stationary phase, which is consistent with previous findings that any sort of pairing seems to be weaker or abolished in S phase. So we next wondered whether there's variation across the genome in terms of pairing. And to look at that, we can go back to this heat map of intergenome interactions. And if you look carefully, you might have noticed this bright spot, which represents the interactions between the homologous chromosomes, which carry the rDNA array. So in cerevisiae, the rDNA array exists as a single, about a megabase long region with 150 or so copies of the rDNA. And this is all packaged at the nucleolus at one end of the nucleus. And because this is roughly the size of the rest of the chromosome, it basically splits the rest of the chromosome into two halves, the proximal half, which contains the centromere, and the distal half, which does not. What we see in this Hi-C data is that the homologous proximal halves seem to interact very strongly with each other, as do the distal halves. And this pairing is much stronger than any of the non-homologous interactions. So we wondered why this might be happening. We had two hypotheses. First, maybe it's sequence dependent. Maybe there's something special about the sequence of chromosome 12. Or alternatively, maybe it's somehow due to just this conformation with the rDNA and centromere tethering. And what I mean is that the proximal halves are both tethered to the nucleolus and to the spindle pole, whereas the distal halves are just tethered to the nucleolus. And these are still unique in that no other chromosomes are attached to the nucleolus. To test this hypothesis, we made a translocation strain in which we swapped most of the proximal half of the cerevisiae chromosome 12 with an equivalently sized piece of cerevisiae chromosome 5. If in fact the pairing is sequence dependent, then we shouldn't see any change in the pairing. But instead, if it's due to this shared tethering, then we should now see pairing between the cerevisiae chromosome 5 and the bayanus 10. And that is in fact what we see. So that suggests that what appears to be mitotic homolog pairing can sometimes be completely independent of sequence. So far, I've talked about homolog pairing at the scale of the genome or a single chromosome. But we have data to suggest that it can occur between specific loci as well. So here I'm zooming into the interactions between the homologous chromosome 13s. And I've circled in white here a region where you can see that homologous loci, which contain the gene Has1, and I'll refer to that as the Has1 locus, seems to interact very strongly in stationary phase, but not particularly so in exponential growth. And this was surprising to us because there was no report of anything like this in the literature. So if you didn't believe me from the heat maps, I'm also going to quantify it here. And so I've labeled the strength of this Has1 pairing with the red line. And the gray distribution represents the distribution of all of the sort of similar comparable interactions. And you can see that in stationary phase, the Has1 pairing is far and away the strongest interaction, whereas in exponential growth, it's fairly typical. So like any good geneticist, we wanted to see if we could make it go away with a deletion. And so we deleted a 20 kb region that spans over 10 genes. Sort of the best guess based on the high C signal. And then we grew these cells in stationary phase to see if the interaction was now gone. And in fact, this deletion brought the strength of the pairing back down to roughly a baseline level. We made smaller and smaller deletions, and eventually we were able to get a single 1 kb intergenic region that was required for this pairing. Now, this intergenic region includes just 2 promoters for Has1 and TDA1. Has1 is a gene involved in ribosome biogenesis, and TDA1 is a protein kinase of unknown cellular function. It's still a bit of a mystery to us as to why this pairing is occurring. We're still actively working on figuring out exactly the mechanism and the implications of this pairing. But our current hypothesis is that this pairing is due to transcription factor binding that's on the 2 homologs that's resulting in either a sort of shared tethering to a third location, such as the nuclear pore complex, or a sort of dimerization. So in summary, I've told you about how we can use high C in yeast hybrids to look at mitotic homolog pairing genome-wide. And we found that it seems to occur even in interspecific hybrids, particularly in stationary phase. It also seems to be particularly strong on the rDNA-carrying chromosomes, cerevisiae 12 and bayanus 10. And it also seems to occur inducibly at the Has1 locus, especially in stationary phase. With that, I'd like to thank all the people who are involved in this work, especially Ivan and Maitreya, who are also both here, and my advisor, Jay Shandari. I'll take any questions. So we've tried moving the 1 kb energetic region to a locus on chromosome 14, and it doesn't seem to be sufficient, at least to create a pairing of similar strength. So it might be that it requires a sort of larger region, but at least this region is required.
So nice talk. I want to understand something. The two yeast strains are really not very homologous to one another, correct? So, and it's well known that in fact homology search gets disrupted when regions are not homologous to one another. So it's the recombination and pairing can't be what's driving some of this pairing, quote unquote. I mean, you didn't say that, but I think you know that.
No,
right?
I'm well aware that it may or may not have anything to do with that. But observationally, it does. The homologs do seem to interact more with each other than you'd expect from just the Rabl orientation,
right.
et cetera.
But the other thing that's maybe worth noting is that you didn't see that in S phase during replication. And that's actually when recombination is going to take place. It doesn't take place in stationary phase because the recombination enzymes aren't, I don't want to say they're not functional, but they don't work as well. So what do you make of all of this? at the end of the day?
So I guess you can attribute possibly some of that to an altered nuclear sort of structure in stationary phase. It's one of them that it can be quite different and is smaller and seen to be misshapen, etc. And then there might be some real signal from examples like the sort of focal pairing that I've seen at the has1 locus, perhaps mediated by, say, transcription factors. I think it might be just a combination of those features.
Have you looked at whether regions that are related through the whole genome duplication have any signal of being paired at all?
I have not looked at that. Thanks. Thank you.
Let's thank all our speakers for this session, and just a reminder for anyone that's interested, come on up and get information about the Ecology and Evolution Conference in Heidelberg.